Hi, I know Lucy has mentioned me a few times already this morning, and there was a questionable photo in your presentation. But I spoke to a few of you at break, so hi to everyone I haven't spoke to. So yes, we do have a speaker, Dr. Sean Walker, who is the Chief Scientific Advisor of our US partner, ViaGen Pets and Equine. And he couldn't actually be here, but he has got a but he has sent us a recording, so I'll play that for you now.
Sean Walker, and I am the Chief Scientific Officer at ViaGen Pets and Equine. And first off, I would like to thank Gemini Genetics for inviting me here to give this talk today regarding cloning and how it can be utilized to save endangered species, but also to protect rare breeds from genetic loss. I guess I'll start off with a little bit of a background about cloning. Cloning is actually a technique in which you take and can make a genetic duplicate of an animal. We like to term it as a identical twin separated in time. And so what we'll do is we'll take genetics from the animal that we want cloned. We'll take a gene bank or a cell sample from that animal. And once we have that, then that's actually saved indefinitely. And then we can actually use that at a later time to do cloning to bring back those lost genetics or the genetics of that animal. I'm here today to discuss a little bit about how gene banking can be used to preserve rare genetics and also how cloning can be utilized to then bring those genetics back into the gene pool. We have been involved with two different projects, both that are using cloning and bringing back lost genetics in the wildlife field. One thing I want to quickly stress in this genetic preservation is the genetic preservation part of this project. None of this work that we are about to talk about could have been done without the foresight to save genetics before they were lost. The San Diego Zoo, one of our key partners, has done an amazing job, and we're one of the first institutions to actually realize this and develop the frozen zoo, which contains thousands of samples from many different species. They have saved this throughout the years, and this will help them and us to preserve genetic diversity if we ever need it in the future. The first project that I'll talk about a little bit about is actually a black-footed ferret project. We worked with a group called Revive and Restore, the U.S. Fish and Wildlife Service at AZA, which is the Zoo Society, and the San Diego Zoo. For those of you that don't know much about the black-footed ferret, it's actually a species of mustelid, and it's native to Central and North America. The black-footed ferret was actually declared extinct in 1979, so a small group was founded in Métis, Wyoming in 1981. From this population, only 18 animals were able to be captured, but only 7 of those ever reproduced in the captive breeding program, which began in 1987. Attempts have continued to be made to reintroduce black-footed ferrets, but have been mostly met with struggles. It's due to its limited number of founders. The BFF population is currently undergoing a genetic bottleneck, and this just means that the amount of genetic diversity within the population is very, very small because of the small number of founders. To help address this genetic bottleneck, we actually worked with we undertook a task to clone a cell sample that was preserved in the mid-1980s. And whenever they actually did the genetic testing of this animal, it was found not to be a relative of the original founding 7. This was done to bring back genetic diversity. As some of you may have saw on December the 10th, 2020, we actually had Elizabeth Ann was born. And so there's a lot of pictures, and you can find a lot of information of her on the web. But the idea is that with these lost genetics, we can now take and open up that bottleneck that occurred. And so Elizabeth Ann and other founders that we'll be cloning in the future will actually be used to broaden that genetic diversity and hopefully help bring that population back. The second project that we have worked on is actually the Przewalski Horse Project, and we worked with that on the San Diego Zoo and Revive and Restore. For those of you that don't know about the Przewalski horse, it's actually a rare and endangered horse originally native to the steppes of Central Asia. It's considered to be the last wild horse and was once extinct in wild, but since has been reintroduced. Similar to the ferret, the group undertook the task of reproducing genetics of the most diverse sample in possession of the San Diego Zoo. On August the 6th, 2020, Kurt, who was named actually after Dr. Kurt Berninsky, one of the original founders of the San Diego Zoo, Frozen Zoo, was born. Kurt is now at the San Diego Zoo Wildlife Park with a female in hopes that he can pass on his unique genetic and also open up the gene pool for the Przewalski horse. As you can see, cloning can be a useful tool to help with bringing back lost genetics of any endangered species or rare breeds, but only if there is the foresight to preserve genetics when available. As in the case with the rare dog breeds, one of the biggest risks is lost genetics. Being prepared for this and aware so you can develop a system to help overcome would be the best plan to put in place. I thank you for your time and hope you have a great day.
So yeah, that was from our US partner ViaGen. I think he explains it quite well how they're using the technologies that they develop for domestic animals to save our endangered species as well. And I do have a few more videos that ViaGen sent to us, just that follow on from the one that you saw through the microscope on Lucy's presentation earlier, and I will hopefully have them up already. So, show you some of the lab work that they do. So we did ask ViaGen to send us this just so we could elaborate and so you could see it for yourselves. So the oocytes or eggs are harvested from a donor animal, and then these oocytes are then placed in a maturation media and grown to the mitotic 2 stage of the cell cycle, and then it's at this stage that they are ready for the fertilization. At this M2 stage, we stain oocytes with a DNA stain, so we can actually see them, and then we use a micropipette to remove the DNA within each of the oocytes. This DNA staining just allows us to use fluorescence under the microscope to basically let them see what they're doing. So yes, that is what you saw earlier in Lucy's video that allows you to see it. And now that the oocytes are devoid of the maternal DNA from the donor, we take the cells that were cryopreserved from the animal that you wish to clone, and then these are then placed into each of the oocytes. These are then given an electrical impulse, which allows the cells, which allows the oocyte membrane and the DNA of the animal you wish to clone to fuse. So the DNA of the oocyte is now the DNA of the animal you want to clone. This is then given a stimulus called activation, which initiates the development of the oocyte into the actual embryo. This embryo is then placed in a surrogate animal between days 2 and 7, following this activation. And then from there, it is just carried out and treated as a normal pregnancy within the surrogate, and the animal is born as it would otherwise would be. And then ViaGen, if it's a dog, ViaGen will look after it through weaning, and they will send you back a lovely clone. So, yeah.